So hello and welcome again. We are in plant biology and I would like to continue my lesson. I first want to give you a short overview over the topics that I'm going to cover today. So I already wrote them down here. First of all, we're still going to talk about transport in plants. So I'm going to talk about an experiment on how you can determine the transport rates in phloem. Let me change the color here again. Remember, the phloem is this vascular tissue that transports sugar from the leaves down to the roots. And then the rest here I'll talk about plant hormones and plant growth. So it is a slightly different topic. Okay. So what I would like to do first is I would like to explain to you, an experiment that they have done to determine how fast the sugar is transported from the leaves to the roots. And the reason why we do this experiment, because it's a very original one, quite a unique one. Okay, so and as a matter of fact, here I already made a little drawing simply to save time a little bit, and I'm going to first explain to you what this is. Well, of course, here horizontally here, that is basically that's our plant. As you can see, the roots here, of course, with water, and it's horizontal. And the leaves here, where photosynthesis happens, is basically there is a chamber here. Okay, we call this a gas chamber made of glass. So it's a glass chamber. Why glass? Of course, because we want to make sure that light, of course, from the sun is able to go in here. But there is gas in there. As a matter of fact, carbon dioxide gas. And do you see the number 14 here? This is a radioactive carbon dioxide. You can actually have that. Okay, so it's radioactive CO2. The reason why it's radioactive is so that we are actually able to see the radioactivity. Okay, so that it's labeled. And then we have over here we have little insects, and these insects are called Aphids. In German, Blattläuse. And they like to suck the phloem. And so what they have done is the following. They have actually put those aphids on the stem here of the plant. And what they do is they have those aphids, they have those mouth parts, and those mouth parts are called stylets. And they basically put aphids here, towards the leaves. So I'm going to just put some aphids, a couple of aphids over here, and they put also some down here. Okay, so those dots, these are supposed to be the aphids. A little bit too small to see. But what they have done is the following. They found the phloem because they wanted to suck the sugar solution. And what they have done is I'm not going to now make a big drawing here. This is the stem of the plant. Okay, so what they have done is they've actually used their stylet and they have punctured the phloem, which is in there. Okay, that's the phloem. And here that's the head of the aphid and the rest of the body. Okay, get the idea. Okay, I'm not gonna draw this now. Yeah. That's they have punctured it, and then they've done something quite interesting. They've actually once they found the phloem and once they started sucking on the sap on the sugar water, they actually broke it off here. Okay, they basically broke it off, and this basically means that the stylets were still in here in the stem, but the rest of the aphid was gone. And what they had then is the following. They had then the stylet. I'm just gonna draw this over here again. The stylet was still in here. Actually, I should draw this upside down. And then the drops started to form, and they started to fall off. So what you had over here is you had at the end only the stylets, and then it started to drip out. Okay, and also over here, and you could collect it down here. Yeah. Because the rest of the aphid was gone, but you used the aphid simply to find the phloem. And then what they've done is they waited until radioactive sucrose, okay, came out here. And this was around, 65 centimeters apart. Okay, so they waited until radioactive C14 sugar started to come out here first, and then they started the timer, and then they waited until it came out here. Okay, and by the time difference, they were able to determine how fast the substances were transported down the phloem. Okay, it's kind of a fascinating experiment. So they kind of used the aphids to find the phloem, and then they removed the aphid, broke it off. The stylet was still in there in the plant, and the sap, the sugar solution was dripping out, and then they simply waited until the radioactive sugar came out first here, and then they started the timer, and they waited until it came out down there, because the substances are transported from the leaves to the roots, and yeah. This is how they were actually able to determine the rate of transport inside a phloem. You should know this experiment. And that's basically a very elegant way, a very interesting way. And I think that's the reason why they've actually chosen it, to show how you can actually design experiments here. Okay, so that's pretty much it, and I'm just gonna move on now to the next lesson. Please do copy this into your nice notebook, okay, and to understand what's going on here. You might also want to write a text here so that it's easier for you to actually study. Okay, so we finished this first thing here. So next one is detecting plant hormones. There is not really a lot to say here, but plant hormones is just a little bit. A short introduction here is, these are of course chemicals. No kidding. You didn't believe that plants also can have hormones. They're of course chemicals, and what they do is just like hormones in mammals, for example, they control a variety of things, but among other things, growth of plants, among other things. Okay, and of course, like hormones, they work at a low concentration. Because the concentration was so low, it's not so easy to detect them. Okay, so what you need is you need analytical techniques, in chemistry. Okay, analytical techniques to detect them. And there are a couple of them that the book mentions here. One of them is called ELISA. That's you use antibodies, so you can check whether the antibodies bind against the hormones. Okay, so another one is gas chromatography. I mean, I don't know if you really have to know this. It wouldn't be, just say chromatography. Okay, that is also an analytical technique. The devices that you use are quite expensive, and the thing is that you have different types of plant hormones or families of hormones, and they all need a different way of purification. Need a different way of purification. So in other words, why am I telling you? It's kind of difficult. They were difficult to study. Okay, because the concentrations are so low. Yeah, that means, okay, so that is one problem. You need special technologies. Okay, low concentration. Then you need a certain equipment, which costs a lot of money. Okay, and the last thing is you need the different hormones. They have to be extracted first before you can actually analyze them. So that means there is a whole bunch of chemistry involved as well. Okay, so you need specific methods. And this kind of made the studying of plant hormones kind of difficult, okay? Because they work in very low concentrations, and detecting those low concentrations is kind of difficult. But those plant hormones actually do play a pretty important role. So for this reason, that was actually it. So that was detecting plant hormones, okay? And the difficulties actually of detecting plant hormones, right? And now I would like to show you a couple of applications. And one of these applications is called phototropism. And this is when a plant grows towards light, okay? And so that is the next topic that I'm going to talk about here. Called this phototropism. There's also something like called a geotropism, where the roots grow towards earth, for example. Okay, but what we talk about is phototropism, and the hormone that is involved here has a name. It's called auxin. Okay, so that's the name of the plant hormone. Yeah, the idea is that plants. If they ask you describe phototropism, don't just write down auxin, but you have to explain what it means first. Plants grow towards light. My handwriting is not very nice here on this tablet. Yeah, I'm not going to rewrite this. Plants grow towards, is supposed to be towards here, light. Okay. Why do they grow towards light? Of course, because at light you can do photosynthesis much better. And so basically what happens is the following. If we look at the tip of a plant, okay, then there is the very tip here. This is the part of the plant that actually grows, and this has a name. It's called apical meristem. The roots also have an apical meristem, but that is the region of the tip of a plant that actually, you have a lot of dividing cells, lots of mitosis going on. And let's say that there is light coming from the side over here, okay, in this case from the right side. Okay, there is light coming over here like this, and what's going to happen is the following. The hormone auxin is going to be transported from the light side into the dark side. So I'm going to make those little arrows, okay? So those arrows here indicate the transport of auxin. So they're pumped away to the shady side. So this means that there is more auxin now, and the auxin I'm going to make as dots, okay? Here on the shady side. Okay, so that is kind of the idea here, okay? So there's a high auxin concentration on the shady side, and what the auxin now does is it causes the cells to become longer. Okay, and as a consequence, what's going to happen is the plant is going to bend over like this, because here on the shady sides the cells are longer. I'm going to exaggerate this now, okay? So you see that the cells here are longer, and here the cells are shorter on the light side, okay? And this causes, this causes the plant to bend over like this, okay? So these are supposed to be the cells. So auxin causes cells to become longer. It elongates the cells. It kind of promotes growth in that sense, right? And therefore, this is possible to happen. Now, of course, people wanted to check this and actually wanted to see if this is indeed correct. So what they have done is the following. Just going to show you here as well. They have essentially they cut off the apical meristem. Okay, they simply cut it off. Okay, so the tip here, okay, was cut off, and they kind of removed it, okay? And then what they have done is they have placed over here, so auxin could not be produced anymore, okay? But then they placed over here a cube, a gel cube with auxin. Okay, and only on one side, okay? What happened? Well, then as a consequence, of course, also here you had more auxin because the auxin diffused all the way down. And then what you had is the following: you had also something like this happening. It's a bad drawing. Okay, so you see that, yeah, you have the gel cube here again. It all started to bend over. So you can trigger this phototropism, also by selectively applying the auxin. So it shows that you do not actually need an apical meristem for this to work. Okay. Yeah, and also you don't need any sun for this to work, because you can actually trigger it like this as well, right? So this is actually an example of how plant hormones can actually cause plants to kind of respond to the environment. In this case, the direction of the sun, right? So so let's go back here again to see what I want to talk next. So that was phototropism. What does indeterminate growth of plants mean, and the growth of the shoot? I think I might cover those topics together here. Okay, indeterminate growth of plants. So I go back again to a foil. How do you spell this? ATE, plant growth. Indeterminate means that there is no maximum size. There is no built-in. Built-in might not be a correct way of saying it, okay? Theoretically no maximum, no limit to plant growth. You're gonna say that's not correct. Trees only reach a certain height. Yeah, but there's a reason why. Because they might not get enough water. Okay, but I mean, humans, for example, animals, other animals, they have a certain size. Where's the limit? But plants keep on growing, and that is basically what indeterminate means. That and the plants grow on two places in the meristem. So that basically causes the plants to grow in size. And the second, and that's actually the one that we'll be talking about most, and the second one is in the cambium. If you remember when we did the cross section of a plant, of a stem, for example, and you had over here the vascular tissues, the phloem and xylem, then in between here you had cells. Not only here in the vascular tissues, but here you had cells that were dividing. And this actually caused the plant to grow into the width. Okay? So these are basically two places where the plant grows. And the thing now is the following is, how does this actually look like? And what I'm going to do now is I'm going to make a drawing, a very, very, very schematic drawing of the shoot. The shoot is the tip of the stem, okay? And what you have here is the following. And I'm going to, I don't know, I don't like this drawing very much. It's not so easy to draw. It's a very schematic drawing. So simply that you a little bit understand how this looks like. So what we have here is the following. You have over here the place where the stem grows, and this is the apical meristem. Apical meristem. So you've got a lot of mitosis going on, cell divisions going on, okay? And that's the region of growth. So growth happens here. But there are also some leaves, okay? And those leaves, they actually grow over the apical meristem like this. Okay, so this might be a little bit confusing because they are actually higher than the apical meristem. So these are the youngest leaves. And you see that they kind of cover the apical meristem. And then what you have is you have also another leaf, and those further down. We go down a little bit here, okay? You have over here some buds, and those buds, they will actually form side shoots. These are buds. And over here, what we have is you have yet leaves that might go quite high. And here as well, maybe not be a very good idea to actually draw it like this. Okay, and this is the tip of the plant. So what you see is, oh, look at this. Oh, isn't this nice? Here we go again. It's always one of these confusing things. Okay. I see, because I put the book on my keyboard, and therefore the book was pressing on the keyboard. Okay. Here we are again. So basically the idea is where's the tip of the plant? Well, the tip of the plant, of course, is here. This is apical meristem. Even though this might not be the highest place, because there are leaves on top of it, but this is a part that's growing. And you see those buds here? They also have apical meristems. Yeah. So what you have is you actually have a plant, obviously, yeah, growing up. Here you have the top one, and then you have side buds forming here and here, and they will also grow. Okay, and then they will also branch off again. And this is basically what we mean with indeterminate. Yeah, and this one will of course also grow higher. Yeah. So this means that you almost have the whole pattern that you have here at the top. Yeah. The pattern that you have here at the top, you have repeating also here and further down, and so on and so on. Okay, so it's kind of repeating over and over and over again. Yeah? So this whole concept is replicated. Yeah. So the idea here is a little bit, it is simply to show you that the youngest leaves, of course, are the smallest ones here. Okay, and as this one grows further up. Okay, those leaves are pushed down further and further, and you have again new leaves growing, and the buds, they will also start to grow. Okay, and then they will again form side buds here, and will start to grow, and so on and so on. Okay, but that is essentially simply to show you a little bit how the tip of a plant looks like. Okay, so and yeah, and that kind of completes the video lesson for right now, and yeah, and we continue with plant science in the next video. Okay, all the best, bye bye.